So in the interest of time, I got a lot of material in this lecture, and I'm not going to be able to cover all of it. In fact, I'm going to cut it off at a place, and I'm going to cover some of it for Friday. Our PSC that we're covering on Friday is relatively short and simple, so I will be covering some of that material on Friday. But I'm going to be giving you all you need to know to do the PSC today. So let's get started. This is obviously a big topic: sex-linked inheritance and sex determination. I know it's a very exciting topic. I actually love this topic because it's really complex. It also draws in a bunch of stuff about the brain. But we got to talk about sex, and so therefore, there's going to be the typical warnings associated with that. But I also want to define sex, right? There's two basic definitions of sex. One is either of the two main categories, male or female, into which humans and most other living things are divided on the basis of their reproductive functions. Of course, the other definition is probably arguably the funner definition. It's just sexual activity, including specifically sexual intercourse. I hate to say this, but we're not going to talk about that much. There will be some discussion, but a little. We're going to be talking about the more boring topic. So the first thing then, if we're talking about sex and like maleness versus femaleness, one way to describe that is to talk about sexually dimorphic traits. Now, many animal species will exhibit sexual dimorphism, and that is that they have different, separate, discrete endpoints, where there's a typical male endpoint and a typical female endpoint with very little or no overlap in between. Now, some animals have extraordinary examples of this where it's very obvious whether you're looking at a male or female based on their appearance. Other animals, it's actually less so. I used to work with a species of songbird, the white-crowned sparrow, where by plumage, you couldn't tell the difference between a male and a female. If you looked at them, you couldn't see. The only way to tell the difference was that if you looked on the inside. And so that's going to be some of those more specific sexually dimorphic traits, such as chromosome arrangement, genitalia, gonads, gametes, being sperm or eggs, and then some secondary sex characteristics. And I would say in humans, there are fewer of those than you might think when you compare that to other animal species, but there are some that certainly do appear to be sexually dimorphic to some degree. So let's get into it. So as far as humans are concerned, right, a typical human will have 46 chromosomes. And there are 22 that are paired off in equal proportions. Those are the autosomal chromosomes. But then we have the 23rd pair that's where things get complicated. And so like most mammals, sex determination in humans is driven by the arrangement of the sex chromosomes. The homogametic sex in humans is the female, and she has an XX chromosome arrangement, typically. And then the heterogametic sex is the one that is associated with XY chromosome arrangement. Those are typically males. And so the answer to this question is what I just said. Homogametic refers to the fact that they have the same kind of sex chromosome, homo meaning same, and then hetero is opposite, therefore they have two different kinds. They have the opposite arrangement, XY. So if you're talking about a species that has sex chromosomes that are very different between each other, that is the heterogametic sex. And in humans, what we have is that females are homogametic and males are heterogametic. And then if a human male and a human female love each other very much, they may have babies. There's a pedigree here. And that the resulting of this mating resulted in both a male and female offspring, largely determined by the fact that the male has XY chromosome arrangement and the female has XX chromosome arrangement. Now, the male generates sperm that is either X or Y because of meiosis, right? Therefore, if a Y type sperm fertilizes an egg, which is going to have X chromosomes, that usually results in the production of a male offspring. And then if the X kind of sperm, half of his sperm is going to be X, ends up fertilizing the egg, that is what usually results in the production of a female. And this is what it's like in humans. Now, there are lots of exceptions to this, including even in humans, as we will get into. And in animals, it's actually not nearly as simple as just saying we have XY chromosome arrangement. It turns out that almost all mammals have this arrangement. Some reptiles Many amphibians and fish also have XY chromosome arrangement where the heterogametic sex is the male. But it turns out there are other animal species where the opposite is true, where the heterogametic sex is actually in females. So all birds are heterogametic, which means they don't have, the males aren't XY. Instead, they are what we refer to as ZZ, and the females are ZW. Now, why do you think that we will have picked ZZ and ZW to describe this in species where the heterogametic sex is the female? Why wouldn't we just pick XY? Yeah.
So there's no
Yeah. Exactly. So we just assign them different letters. So initially we called them X and Y in mammals. Then we discovered in other species that they have the opposite. And it would be too confusing to say, oh, well, female birds are XY. We just pick then the letters that are on either side of X and Y. We picked Z and W, which is on either side of X and Y. So therefore, we call them ZW type species. That's where the female is then heterogametic. Okay. So then this is then the arrangement of when you talk about birds. All snakes have this arrangement. They're ZW species. There are many fish that are like this. Some amphibians are like this as well. And then it turns out that even in mammals, there can be differences. There are some species of mammals, and the more mammals that we look at, the more exceptions we're learning about, where it's actually polygenic, where it's not just that there's a single sex chromosome, and it turns out that, like in this particular species, the African pygmy mouse, there is a sex chromosome arrangement, but then there's also another sex determining factor on a separate chromosome that is not part of the sex chromosomes. And then, so that is now referred to as W, and therefore it's kind of complicated. So it turns out that in males, there's only one genetic type of male, but there are 3 genetic types of females because of this extra sex determining factor on a different chromosome that is not a canonical sex chromosome. This is known as polygenic sex determination. Yeah. Yeah, so insects, it's also highly varied. Turns out fruit flies are XY, but then Lepidoptera, which is going to be moths and butterflies, tend to be ZW. So it's also highly varied. And then there are other species where they don't actually really have very clear sex chromosomes, but they do have sexes. But then that is largely determined by gradient of temperature that they experience during development. So temperature can also have an influence. I'm not even going to touch that because I got way too much to talk about as it is. Some of that's described in the textbook. Yeah.
So
Yes. So I don't think so. I haven't investigated this case in particular. The citation is right here. It is publicly available. I encourage you to take a look if you want to learn more about it. I don't exactly know. I think that that would be the case, that you're more likely to have more females than males. And there are times and there are certain kinds of species, especially with fish, where it's more conducive to having fewer males than females because of the social structure that the fish have, because they tend to have harems. And that's a thing. And then there's also sex switching that happens in fish. So they can change from, say, female to male as adults. So nature's wild, and as we're going to learn about. So speaking of fruit flies, now I want to talk about X-linked traits, or sex-linked traits, I should say. So the first discovery of this, of a sex-linked trait was eye color in fruit flies. This was discovered, I think, in 1905. And basically, some researchers, they were mutating flies. They found a fly that had, and it happened to be a male, which is kind of important, that had white eyes. So they had mutated, apparently, a gene for the red eye, and that resulted in flies that have white eyes. But they happened to see that it was associated with a sex-linked relationship because of the way the Mendelian inheritance worked out. So we're going to walk through this a little bit. So if you were looking at a red-eyed fly, you may have a female that is homozygous for the red eye version of the gene. They have 2 red alleles, so they're homozygous for red. They could be heterozygous because they may have the red gene and then the white version of the gene, the mutated version. But then if you're talking about a male, because this gene is on the X chromosome in fruit flies, If it's red-eyed, it means that it has only one copy of the red version of the gene, right? Because the Y doesn't have any eye color gene because it's only on the X chromosome. But because males in fruit flies, just like say in humans, they will have some genes that are only found on X chromosomes. They only get one shot at it. They don't have two to work from. So you cannot have a male that is heterozygous for a sex-linked trait in this kind of animals that have this chromosome arrangement of XY chromosomes. Does that make sense? So that would be the three versions of genetics that we would see if you're looking at a red-eyed fly. For white-eyed flies, there's only two versions. If we're looking at a female, she will be homozygous for the white mutant. So both chromosomes would have to be white. And what does that mean? Is the white gene recessive or dominant? It's recessive, exactly. This is clearly indicated by the heterozygous, because the heterozygous has fully red eyes, and therefore, but we could do a genetic test to see that it has the white mutation. If we're looking at a white male, that means it has one chromosome that has the white gene on it, but it doesn't have a red gene because it can't have a red gene in addition to the white gene because it is only allowed one X chromosome, right? Because that second place is taken up by the Y chromosome. So this is where you can kind of see that maybe inheritance can get a little complicated when we're talking about sex-linked traits. Okay, so let me walk through this a little bit. So let's take an arrangement where we have where we cross a homozygous red-eyed female with a white-eyed male. So this female has two copies of the red gene, and we've got a white-eyed male that has the white version. If we do a cross, the females are all going to be heterozygous for the trait. They will all have red eyes. Now, why is that the case? Why is it all? All of them are going to have red eyes, but they're all heterozygous. Yes.
Because you need to be homozygous for the white.
That is true, but why are all the females here heterozygous for white eyes? Why isn't it like half are homozygous for red and half are homozygous for white? Yes, in the back. Exactly. They don't have a red X chromosome to donate. They have one chromosome to donate. So the males are determining the gender of the sex by either having X sperm or Y sperm, just like it is in humans. But if they have the white chromosome, and if we have a female that we know is homozygous for red, then all the females have to be heterozygous white. They have to be. Because they're getting the white chromosome from dad and the red chromosome from mom, because she's homozygous. And therefore, whether you get the left red chromosome or the right red chromosome from mom, it doesn't matter because they're both red. So you're getting definitely one red gene from mom, for sure. And since it's dominant, that means all those females have to have red eyes, but they are all heterozygous. Now the males are all red eyes. Now why is that the case? Why isn't it like half and half? Yes.
Because if it's male, then they will 100% get the red from the X chromosome from the female, and 100% get the Y chromosome from the male.
That's right. And if you have a male fly, it means it got a Y chromosome from dad. It didn't get an X chromosome. So it can't get the it can't get the white version of the gene from dad because it can only get the Y chromosome. You can't have a male getting the white version of the gene from dad because you're getting the Y chromosome from dad, right? So therefore, it cannot be that you can get any white-eyed males from this kind of cross. It's just impossible. They will all be red, and therefore they are, they've got the red version. Now, if you do another cross, I'll let you work it out, like how this all works out. But essentially what happens then is, so you do a cross between these two individuals, you're going to end up with males that half the males are going to be white eyes and half the males are going to have red eyes, and all the females are actually going to have red eyes. And I'll leave you to figure that out, precisely how that actually works out, because on the PSE there's a way of we're going to be asking about X-linked traits. So There's another version of this where you can say, like, what happens if we cross a homozygous white-eyed female with a red-eyed male? So if we're talking about a red-eyed male, that means it has one red X chromosome and then it has a Y chromosome. And if it's a white-eyed female, like we don't even need to test to see if it's homozygous. If it's white-eyed and it's female, it has to be homozygous for that. Hold on a second. Just double check something here. Okay. All right. So therefore, all the males in the next generation, oops, right here, all the males are going to have white eyes because they're getting the X chromosome from mom, and she has two versions of the Y chromosome. So all the males are going to be white, and then females are going to be heterozygous for the trait because they're getting one red X from dad, but they're getting a white version from mom. So all the females are heterozygous. And then this is what it looks like in the second generation. So, and I'll leave it to you to figure that out as well. Because I need to continue talking about pedigrees and sex-linked traits. So I want to walk through like how we would work our way through a pedigree. So this is a typical way of having a legend to describe a pedigree. A pedigree is when you look at like a family tree, essentially. But a pedigree would allow us to follow traits through the tree. So females are going to be shown as circles, males are shown as squares. If we're following a trait, and if we're saying that someone is affected, they would then be like filled in with some sort of color. And in this case, we're looking at blue. If parents have mated, then by definition, in order to be parents, they will have mated. There'll be a line connecting between them, and their offspring will be shown below, like so. So in this case, this would be a cohort of full brothers and sisters that are related, where we got two brothers and two sisters. Twins are shown like this, and then generations are indicated by I, double I, or triple I. Okay.
They're related.
Yes.
Okay.
That's what that means. That does happen more often than you might think. Yes. All right. Fortunately, there are no examples in the pedigrees we're going to be looking at. So, okay. So if we're talking about an autosomal recessive trait, and it it is often the case that those recessive traits will skip generations. And so, like an example of this, and hair color is not this simple, but one example is like red hair color. Oftentimes, people describe that as like something that skips a generation. So we'll sort of describe like how that actually works, looking at a pedigree, because you can figure out some of these traits if they have like simple Mendelian inheritance and there are autosomal like recessive traits. You can kind of figure out if it's a recessive trait by looking at a pedigree like this. So in this instance, we have a dad. Let's say he has red hair, and if we know that like red hair is like a recessive trait, as it typically is, therefore he would be homozygous for like the red hair version for hair color. Mom could be heterozygous or homozygous. Hard to say. In the next generation, we end up with 3 children that don't have any red hair. That would put evidence in favor that maybe mom was homozygous for not having red hair traits, and dad was homozygous recessive. That would indicate that all these children are heterozygotes, right? They're all carrying the red version, so they are carriers. Now, this young man here, Bob, meets Sally. They, fortunately, there's no double line here. They're not cousins, so it's not consanguineous. They get together. They have a bunch of kids. They've got 6 kids. They've been busy. Okay, so and then this is all their kids, and it turns out 2 of the kids have red hair. Now, what does this say then about Sally's parents, the grandparents, her parents? It's written on the slide, so you could probably just read it.
Dominant recessive.
Yes, that at least one of them had to be recessive for red hair. If they have kids that all of a sudden have red hair, and neither of them have red hair, that must mean, and especially if like one of the 4 grandparents had red hair, that must mean that on the other side, that at the very least one of them was heterozygous for this trait. And so oftentimes autosomal recessive traits will skip generations because of this kind of thing that occurs. And so a lot of times it happens that way. And this is also true with sex-linked traits. And then dominant traits, I mean, I'll leave it to you to walk through this, but dominant trait, you'll tend to have more kind of more obvious traits that show up in every single generation. And it'll tend to pass through a generation over time consistently because it's a dominant trait. Whereas recessive traits, a lot of times it will skip generations in order for it to be observed. And that's why we talk about generation skipping. And now you know why. Okay. So now X-linked traits for color blindness is provides the opportunity for traits to then be passed in a similar way to like the way eye color is in flies. So in humans, it is relatively rare to find a woman who is red-green colorblind. They, of course, exist, but it is much more common in men. Around 1 in 10 men have red-green color blindness, where I think that in women it's like less than 1%. And so in this pedigree, what we can see here is that grandma, I should say, has red-green color blindness. And what that means, and if we know that it's a recessive trait, she happens to have this, she must be homozygous for this trait. In the next generation, she has 4 kids. Both of her male heirs have the trait, and neither of her female heirs have the trait. Now, her daughters don't have the trait, but the sons do. This is an indication almost immediately that this could be an X-linked trait. And the reason why is because if mom is homozygous for this trait, that means both of her genes are affected with the mutation. So in some ways, she's just like the white-eyed female fly. By necessity, her sons have to be have the recessive trait if it is an X-linked trait. Every single son that she has is going to be red-green colorblind because she only has the mutated version of the gene to pass on to her sons. But her daughters are going to end up with having not the red-green colorblindness because if dad is not red-green colorblind. And by necessity, all of them are going to have normal vision because dad has a normal version of the gene. Okay. So again, following the same exact pattern that we saw with the flies, we can see the same thing happening in humans. Now, in this case, this daughter then meets some young lucky man, and they have a bunch of kids. And it turns out one of the sons has red-green color blindness. What does that say about number 5, the daughter? That she's heterozygous for the trait. Yes. So she can have sons that don't have red-green color blindness, but if she has enough sons, about half of them will be red-green colorblind. And so now you can see why some traits are sex-linked and why they may be more highly associated with some sexes than others. And red-green colorblindness is an example of that. Okay. Another example of this is hemophilia. I'm not going to go into the details of this pedigree. I encourage you to take a look at it. But in the interest of time, I'm just going to go through it relatively quickly. But as you know, many gene systems tend not to be like fully dominant or recessive. That a lot of times we have more complex types of expression and organization. And hemophilia is one example of that. So if a male, it is an X-linked trait, and if a male has inherited the hemophilia gene, he will have hemophilia. It is the case that a carrier for the hemophilia gene, so she's the mother or the grandmother might be heterozygous for the gene. Many times those females will have some traits that are hemophilia-like, like easy bruising, prolonged bleeding, very heavy menstrual cycles, but they are milder than they are in males. So if she is homozygous for the trait, you can have a female that is hemophiliac, but it's often the case that it's going to be males that are carrying this trait. Okay. So similar sort of relationship. But the fact that it's not like fully hidden in females tells us some interesting things about the way the X chromosome works, which is I'm going to be talking about in just a few slides. So, but in this family, there's also the Romanov family of Russia, which had one male heir that had hemophilia because of the X-linked trait, which you can follow through as well. Now, I want to talk about how sex determination and sex chromosomes can kind of go awry, and how there can be deviations from the typical pattern of either XY or XX. An example of this is a form of aneuploidy called Klinefelter syndrome. This is when an individual has 2 X chromosomes and 1 Y chromosome. So an individual with 2 X chromosomes and 1 Y chromosome is typically assigned male at birth. They tend to have sort of certain physical features like being very tall and long arms and legs, unusually long arms and legs. They tend to have some reproductive problems in their reproductive ducts, like internally. Their testes tend to not work very well, but they also tend to have some feminine features as well. So they tend to grow breasts. They tend to have certain female-like behavioral traits. They also tend to have rounded hips. So this tells us that, like, okay, it turns out that maybe the numbers of X's is kind of important to some degree. And it actually, there's an kind of the flip side of this condition is known as Turner syndrome. This is when an individual has only 1 X chromosome and 0 Y chromosomes, okay? So these individuals tend to be phenotypically female because they don't have a Y chromosome. They tend to have female-type genitalia, but they tend to have ovaries that don't fully develop completely. They also tend to have short stature, and they tend to be cognitively impaired. So aneuploidy can result in like some specific physical phenotypes that make it pretty clear that like the number of chromosomes is important, especially when you're talking about sex chromosomes. And so there are various ways that that X chromosome gene content can be controlled. And in mammals, there's often the case that many cells will have suppression of 1 X chromosome, allowing genes to be expressed from 1 X chromosome, leaving the other X chromosome to express the genes that are on there. And this is known as X-inactivation. You can do a test where you can take like a cheek swab from a mammal and then do a staining procedure to look at the nuclei. And if that mammal is XX in its chromosome arrangement, you will see something called a bar body in the nucleus. This is the bar body right here, taken from a female. And if it's a male, it won't have a bar body. And what this is an X chromosome that's within the nucleus. So, right, all this is chromatin that's kind of spread out. Remember the chromatin domains I talked about. But there'll be one, if you're talking about a female, where in many cells, it will be condensed and kind of hidden away. And this is because, in general, there are many sort of cell types where you don't want too many genes being expressed from the X chromosome. Too many of them end up doing goofy things, depending upon the tissues that we're talking about. And this doesn't have like a whole lot of specific control, as we will see in a moment. So these are referred to as bar bodies. And there are some individuals who actually end up with like 4 X chromosomes and no Y chromosomes. How many bar bodies might you expect in an individual that has 48 chromosomes, including 4 X chromosomes? You're saying 4. You're saying 3. Do I got 2? Do I got 2 here? 2? What about 1? 1? No, you got 2? Okay, so we got 4, 3, 2. Turns out it's 3, right? Because you want at least 1, right? So you will see, you can do a test to determine if that person has XXXX. That's that 4 X's. Those individuals will typically have 3 bar bodies because it's trying to control for having that many X chromosomes. So, but it doesn't fully work. So these individuals tend to usually have developmental problems and other issues go on, just like Klinefelter syndrome and Turner syndrome. But the way this X-inactivation works, it happens in the developing embryo. So in the initial embryo, right, you're at the single cell stage. Both X chromosomes are going to be available if we're talking about an XX individual. And then as cells start to divide, it'll still be, both X chromosomes are still producing gene content and still making the things. But eventually what we start to see is that as the cells are dividing and as the cells are turning into different tissues, some X's get turned off in some of the tissue. And which X gets turned off, whether it's like the one that came from like grandma or the one that came from grandpa, is random. It turns out that it's completely random. So obviously, if one came from mom or one came from dad. So in some tissues, it might be that like it's mom, and in other tissues, it might be that it's dad. This randomness is thought to explain certain kinds of traits that we see in many mammals, and a great example of this is calico cats. So typically calico cats are just female, right? You can't have like a male calico cat, and a calico cat is going to be the kind of cat that has like this patchwork pattern that looks very random on their skin, right? They'll have like brown and gray and maybe some white, like, and it's very, very patchwork. It's thought it's because that the hair color trait is X-linked. And because they're getting, say, one hair color from mom and another hair color from dad, as the skin is developing and as they're assigning, like, oh, you're going to express this gene or that gene, some genes are being inactivated on one chromosome from dad and the others are being inactivated from mom, and sometimes it's expressing both. So therefore you get this patchwork pattern because it's random. And so this randomness that occurs allows for traits like that to emerge, and where you only see it in females and not in males, because males, they only have one version, they only have one X, so they don't have X inactivation. You're only going to see it in females. Does that make sense? Okay. So there's a process by which this seems to occur. It looks like that there's a gene called XIC that's found on the X chromosome in mammals. And that you need this gene that allows them to pair off, and that allows for one chromosome or the other chromosome to get inactivated. Because if you break the XIC gene, basically what happens is that the inactivation becomes very chaotic. Some cells, you still get both chromosomes that get activated. So neither chromosome is inactive. Sometimes it's one or the other, or sometimes it's both chromosomes being inactivated, which obviously isn't good because you need some X-linked genes, because now this is an individual that doesn't have any X genes turned on. So you need that XIC gene to work. So that's an important thing. And then if you add the XIC gene to a chromosome that is not an X gene, now you mess up the cell's ability to do X chromosome inactivation, because now some of the X chromosomes are linking up with this random gene that like might be by chromosome 6 or something like that. It's not the right chromosome. So then you break the system. So the XIC gene is really key to this process. Okay. All right. That's mostly what I got to say about chromosomes, although we'll be talking about more about it a bit. Any questions before I go on to the next part? All right. It's a lot of material. I know. That probably could have been a whole lecture right there. Right. I know. Well, get ready, because there's more coming. It's a big topic. So as we said, if we're talking about sexual dimorphism, chromosomes are things that are sexually dimorphic, but there are things like ovaries, vaginas, estrogen, and eggs that we find in females typically. And in males, it's going to be things like testes, penises, androgens, and sperm that are associated with males. And so these are also highly sexually dimorphic traits associated with either one sex or the other sex. Now, how do we get male or female? In mammals, it typically works through what's called the SRY gene, okay? So this is a gene that is found just on the Y chromosome and not on the X chromosome. This is a gene that induces the bipotential gonad to turn into a testis, okay? So in the developing embryo, there is a part of the embryo that starts to make a gonad. It is called bipotential because it could go either female or male. It doesn't really have the traits of either one or the other quite yet. As it's developing, it's just kind of this streak is what it's called. It's a gonadal streak or bipotential gonad. Now, if the embryo has XY chromosomes and if it has a functional SRY gene, that SRY gene gets turned on, and then and therefore it starts acting on the cells that are within the bipotential gonad. And it turns that bipotential gonad into a testis. If there is no SRY gene, it ends up just continuing its development and it defaults towards an ovary. So the default pattern for a fetus, including human fetus, is towards female. But if there is a Y gene present, it will turn on the SRY gene, causing the bipotential gonad to turn into a testis. So this is the first sort of big step, the genetic step that then causes developmental changes that ends up determining sex in humans. Now, SRY is we call it the testis determining factor, okay? It is also a transcription factor. Can anyone tell me what a transcription factor is? So the SRY gene makes a protein called SRY. That protein is a transcription factor. What is a transcription factor? What does that mean? It's literally written on the slide. You could probably just read it and I wouldn't know the difference. So want to give that a try? Sure.
It binds to promoter sequences that regulate.
Man, I couldn't have said it better myself. Perfect. Yes, so SRY protein is a transcription factor that binds to promoter sequences that regulate the expression of many genes. Have we seen another type of gene that is a transcription factor? In the lac operon, for instance. Yes, you're nodding your head, David. What is that gene? Or the protein? Starts with an R, the protein. Oh, come on. You guys forgot all this stuff already? The repressor protein, right? That's a transcription factor that acts on a promoter sequence that affects the expression of genes. So you can call that a transcription factor as well. SRY is a similar thing. It creates a protein that will then help promote or suppress the expression of various genes. One gene that it turns on is called SOX9, which is also a transcription factor. So now you got transcription factors causing the expression of transcription factors, which then turns on genes that are testes specific. So these are genes that are going to turn the bipotential gonad into a testis. And it also suppresses, that's what these little block lines mean, the expression of genes that would turn the bipotential gonad into an ovary. So it then diverges. And this gets really complex. So this is taken from a different figure showing that SRY turns off ovarian genes. It turns on testes genes. It also turns on SOX9, which acts as its own transcription factor, turning off ovarian genes and turning on testicular genes, causing the bipotential gonad to turn into a testis and to deviate away from its default state, which would be an ovary. Okay? So what this means is that in the developing embryo, if there's an SRY gene present, then the gonad turns into a testis. And what that then means is that the testis is free to do what it needs to do, which is to, in mammals, produce a whole bunch of testosterone in utero. So the testes start to produce a whole bunch of testosterone, and then that masculinizes the rest of the fetus. So the testosterone then goes across the fetus, masculinizing the tissues. That includes getting rid of things that are female-like, such as like fallopian tubes and things that are like that would turn into a uterus. It masculinizes things like the clitoris, turning the clitoris into a penis. So the clitoris is actually just like a tiny little penis that in males gets turned into a penis. The vulva get closed up and sealed shut. That turns into the scrotum. And then the testes, which normally would be the ovaries, descend and then go into the scrotum. And it's testosterone acting on all those tissues to do that. But that only can happen if you have an SRY gene that doesn't. Okay? So now we're masculinizing the fetus. Okay? So that is happening in utero. That's what's being shown here. So in utero, we have this surge of testosterone that occurs in males. The level of testosterone we see in the developing human fetus is equivalent to what is seen at peak puberty in males. So it's a lot of testosterone. If you've ever been around a male at peak puberty, you would know. But then it wanes, and then at birth, basically testosterone is largely shut down. And then throughout juvenilehood, there's a hiatus on any sort of masculinizing or feminizing effects of sex steroid hormones. Once puberty is reached, then the hormones get turned on. There's a process by which this occurs, which I'll explain in the next slide. And then in males, if they have testes, they produce testosterone, and in females, they produce estrogen from their ovaries. And then the males will then have the testosterone acting on their male-like tissues the male-sensitive tissues to masculinize those things, such as causing facial hair and stinkiness. And then females, they have feminizing effects, such as like breast growth and changes to hip structure and that kind of thing. So I'm going to kind of just briefly talk about this. I want to say more about it, but I just don't have a whole ton of time. One thing about the way hormone secretion is controlled is that it's controlled by the brain. The pituitary releases factors known as gonadotropins. These gonadotropins are known as luteinizing hormone and follicular stimulating hormone. This is true in both males and females. But the nature of the timing of the release differs between an adult male and adult female. That's what allows ovaries to have a monthly cycle, whereas for testes, they're constantly producing testosterone and they're constantly producing sperm. But it is both the same factor. It's LH and FSH that act on their receptors in the ovaries and the testes that cause them to release testosterone or estrogen, depending upon which gonad we're talking about. Okay. So there's a way that this gets controlled, which I'm going to skip because in the interest of time, because I want to talk about how hormones work. Okay. So if we've got androgens in males and estrogens in females, androgens, hormones work with a lock and key mechanism, right? There's going to be a receptor for those hormones. The hormones go throughout the body, and then it will act on, say, male androgen-sensitive tissues. An androgen-sensitive cell will be expressing androgen receptor If it doesn't have androgen receptor, it's not androgen sensitive. Androgens may enter the cell and they will bind to their receptor. So they will bind to the androgen receptor. If we're talking about a typical female, she will have elevated estrogens that will enter the cell and it will bind to estrogen receptors. Now, there are cells that actually have both androgen and estrogen receptors in both males and females. So, yeah, so it's not, this isn't an X-linked type thing. So, androgen receptor will bind to, androgen binds to the androgen receptor. So testosterone binding to the androgen receptor. It creates an androgen receptor androgen complex. So this hormone receptor complex now binds to parts of the DNA known as either androgen response elements, AREs, or estrogen response elements, EREs, okay? So those are basically promoter sequences, basically the same kind of thing that we talked about for the lac operon. Now that we have this complex binding, that then allows for the transcription of either androgen sensitive or estrogen sensitive genes that then changes the nature of those cells, causes the cells in your face to now sprout facial hair, causes the muscles in your and the ligaments that make up your vocal box to become thickened in males. That is androgen sensitive tissue, causes those cells to produce, say, more collagen or something that then thickens the tissue, or causes the hair to now sprout from your face because it's hitting those skin cells to say, oh, okay, well, now we need facial hair because now we got to be a man now, I guess. Okay. So that's how that works. And it's a genetic mechanism that uses hormones binding to a receptor complex that then binds to either an androgen response element or an estrogen response element. Okay. Does that make sense? All right. I know it's a lot of material, but it's all the stuff that you've learned. I'm just adding a hormone part to it. So let's talk about this and how this affects mammals and the way that they develop. So we can divide hormone control over behavior into two phases. There's organizational phases and activational phases. So when hormones are acting in utero, or just postnatal in the case of rodents, just barely postnatal, that we would say that it's acting in an organizational way. It is changing the way the fetus looks to either be more male-like or more female-like. And if we're talking about mammals, it's testosterone that's doing that changing for the most part. So in rodents, the testes begin to secrete testosterone around embryonic day 18. So this is happening in utero. Rodents have a very precocious birth. If you've ever seen a little baby rodent, you see that it looks like a little maggot, like a little pink maggot. So they're very tiny compared to like a human baby, and so therefore their sensitive period to testosterone overlaps birth. So it's pre and postnatal development, but it's a period of around like 12 days when they're sensitive to the masculizing effects of testosterone, and then they lose that sensitivity to testosterone. So basically what happens is that a male that has normal testosterone development will then be releasing luteinizing hormone and follicular stimulating hormone the way a male normally releases it. It'll just kind of go up and down several times a day, causing the gonads to make whatever hormone they're making, and in males it's going to be testosterone. And so they have a constant sort of elevated level of testosterone. In females, they will have also just some luteinizing hormone, but they'll get this big surge of luteinizing hormone that occurs about once a month, which is related to menstruation or menses in humans, right? So you get a big surge, and that's a difference, just a sexually dimorphic type thing of the way hormones are regulated between males and females normally. Now, if you have a rodent that is castrated during this phase, so it's born at postnatal day 0, you take the testes out. So that castrated male is allowed to grow up and be an adult. As an adult, it has LH surges that are like a female. It has big surges acting as if it is like wanting to have menstruation. But it doesn't have testes, right? Because we remove them. It doesn't have ovaries, so it can't really do menstruation. It's also genetically male, but is now acting like a female in some ways, at least as the hormones are concerned. If you have a female that is given testosterone during this sensitive period, so you give her testosterone during this period, she ends up with a hormone profile that is not female-like, and instead just has these little pulses of LH that occurs daily. She never ever will have a big LH surge. So this is what happens as an adult for her hormone regulation. Now there's also some interesting things about their behavior. So if you have a genetic male that has normal development, they have normal testosterone that occurs during this development. As a male, it will typically want to mount females. So he will find a female and try to mount the female. If he was castrated during this phase, he will do what's called lordosis. So he will stick his hindquarters up like a female would, and then he will allow males to climb upon him and try to penetrate him, even though he doesn't have the parts. He's got a penis. If you have a female that has normal development, so she was not exposed to testosterone early on, she will do the lordosis, stick her hindquarters up the way a typical female will, and allow males to climb on top. But if you give a baby female testosterone during the sensitive period and allow her to grow into an adult, she will show male-like sexual behavior, where she will climb upon the backs of females and try to penetrate them, even though she can't because she doesn't have a penis. And this is how you can get very radical reorganization of behavior by just kind of having different kinds of expression profiles. Since this is a change in behavior, it necessarily means that there's probably a change that's happening in the brain. So it turns out there aren't too many sexually dimorphic features of the brains in both in mammals, but one part of the brain in rodents that is highly sexually dimorphic is called the sexually dimorphic nucleus of the preoptic area. This is an area of the brain that it does control the release of sex steroid hormones. So these are filled with neurons that control the release of LH and FSH. And it turns out that if you are looking at the brain of a female rat, she will have little SDNPOAs. So this is shown here. This is the SDNPO here. It's very, very tiny. And in males, it's big. It's much bigger. There's a lot more cells. They're bigger. So in males, they're big. In females, they're very, very tiny. Now, if we do that experiment again, where we repeat like either castrating or giving females testosterone, we see some unusual things happening in the brain. If you give a female rat testosterone during that organizational phase, during that sensitive period, she ends up with an adult SDNPOA. So as an adult that looks male-like. It is big, like this. So this is a genetic female with a male-like SDNPOA. And if you castrate a male rat during the sensitive window, and if you look at the SDNPOA in the male rat, they have female-like SDNPOAs. So you can reverse whether it looks male-like or female-like by just changing whether they get exposed to testosterone or not in these early phases, even though they're both XX or XY. It doesn't matter. The chromosomes actually don't matter in this case, because here we are manipulating the hormones that they're exposed to early on. And you can reverse their sexual behaviors just by that simple manipulation. But it has to happen during that critical window. That critical window closes. If you do it as an adult, you don't get the switch. So this is largely things that are happening in utero, and probably in humans as well. All right, so there's one last thing I'm going to talk about for today. And I'm going to be finishing up this when we do PSEs on Friday, for which there's PSEs that are due for Friday, a PSE. As I mentioned, there are songbirds that where you can't really tell the difference, but there's some species where it's pretty obvious. An example of this is the zebra finch. So males have these orange cheek patches, they have stripes that are on their chest, and females don't have orange cheek patches, they don't have stripes. The other thing that's interesting about most songbird species is that it's primarily or entirely just the male that sings, and there are brain circuits that regulate singing behavior. And those brain circuits, we call that the song control system. They're larger in males than they are in females. And this is a picture taken from a paper that I wrote many years ago now. But you can see that this brain area is much bigger in males than it is in females. So there are big sex differences in just the singing parts that correlate with whether they sing or not. I'm going to skip ahead this slide, and I'm going to show you an example of what can happen when the genetics gets kind of screwed up. So this is a paper that's about one bird in particular, just a single bird, and it was published in a very high-profile journal. This is a gynandromorph zebrafinch. So this is a zebrafinch that is half male on one side and then half female on the other side. You can see if you look straight down the middle, you can see the zebra stripes end at the midline. You can see that it has an orange cheek patch here, but no cheek patch on this side. And you can see that this is the right side of the bird here, and this is the left side of the bird. And when they killed this bird and then looked at the inside, they saw that the right side had testes and the left side had an ovary. So there was one testis and one ovary. So again, splitting in that, clearly that some of these things are being genetically determined. And then, as I mentioned, like the birds have the ZZ-ZW chromosome arrangement, meaning that the female side is the heterogametic sex. When they look at the brain, so this is a slice of the brain here. So this would be the left side of the brain. This is the right side of the brain. This is the cerebellum. This is cortical-like structures here. When they stain for just the Z chromosome, they see that all the cells have Z chromosome, which is what you would expect, right? Because males are going to have ZZ, right? Because they are the homogametic sex, and females are ZW. So of course, you're going to see Z's on both sides. But if you just stain for W, so there's a way to do this stain where you can only stain for W. The right side of all these brain sections was absent of cells for the most part. There were a few cells right near the midline, but very few cells at all. But the W chromosomes on the left side were all the cells had W chromosome. So this was an animal that probably emerged from 2 eggs that were combined in together, where one egg was Z and the other was W. And then it ended up fused, where then one half of this animal just ended up developing male, and the other half of this animal ended up developing female because of that split. And so this kind of gynandromorphism is a very rare example, but it can teach us an awful lot about how genes and or hormones may end up affecting how we get sexually dimorphic traits in animals. So I'm gonna leave it there. I know there's a lot of material to cover, but it's a very complicated topic, and I still have more to cover. So I did record this, so I'm going to put this up on YouTube so that you have the material. And we will see you on Friday.